Yeah. Hello, everyone. Glad to see you all again for the second session. So, while I was teaching the Morgan's X-linked inheritance, according to Morgan, what he says, the white eye color, what is there? Eye color gene is there on X chromosome, he says. So, how we will prove it? We have to prove it mathematically. So, while solving last time, no, I made a mistake there because while solving, we have to assume. ನಾವು ಏನಂತ ಅಸ್ಯೂಮ್ ಮಾಡ್ಕೋಬೇಕು ಅಂದ್ರೆ ಆ ವೈಟ್ ಕಲರ್ ಇಸ್ ಪ್ರೆಸೆಂಟ್ ಆನ್ ಎಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ ಅಷ್ಟೇ ಅಂತ ಅಸ್ಯೂಮ್ ಮಾಡಿ ನಾವು ಅದನ್ನ ಬರಿತಾ ಹೋಗ್ಬೇಕು. ಬಟ್ ಐ ಕನ್ಸಿಡರ್ಡ್ ಆನ್ ವೈ, ಸೊ ವಿ ಗಾಟ್ ಕನ್ಫ್ಯೂಸ್ಡ್ ಇನ್ ಬಿಟ್ವೀನ್. ಸೊ ಲೆಟ್ ಅಸ್ ಸಾಲ್ವ್ ನಾ. ಓಕೆ, ಫೈನ್. ಸೊ ಸಾಲ್ವ್ ಅಲಾಂಗ್ ವಿತ್ ಮೀ. ಪಿ ಅಂದ್ರೆ ಪೇರೆಂಟ್ ಜನರೇಷನ್. ಸೊ ಪೇರೆಂಟ್ ಜನರೇಷನ್, male as well as female. So in the male, he selected white eyed, which is recessive. So ಇಲ್ಲಿ ಹೇಳ್ಬಿಡ್ತೀನಿ, white eyed ಅಂತ ಅಂದ್ರೆ we will represent it as, just a minute. White eyed ಅಂದ್ರೆ we will represent it as W. Red eye ಅಂದ್ರೆ we will represent it as W plus. So he considered white-eyed male, so XWY, okay, and crossed with red-eyed female, XW plus XW plus. So during gamete formation, what happens, guys? During gamete formation, XW we will get one gamete, Y we will get for some gametes. Again here, all the gametes will get XW plus only. So after fertilization, we got XW plus XW and XW plus and Y. XW plus XW, XW plus and Y. Just a minute. XW plus. So we got one XW plus XW. So this is red-eyed female. Next we got again XW plus XW. So this is also red-eyed female. Next we got XW plus Y, red-eyed male. After that, sorry, my screen sensor is not working properly. So XW plus XW is a male crossed with XWY. This is a white-eyed male. So what we got, the gametes here, XW plus is one gamete, X raised to W is the other gamete. Then here XW plus is one gamete and Y is other gamete. Now, when the cross happen, XW plus XW, this is a female red-eyed. Next, XW plus XW, this is a female red-eyed. XW plus Y, just a minute. Okay, you just help me now. Cross between XW plus XW crossed with XW plus Y plus Y. ಇದನ್ನ ಕ್ರಾಸ್ ಮಾಡಿ ನನಗೆ ಗ್ಯಾಮೆಟ್ಸ್ ಮತ್ತೆ ಆಫ್ ಸ್ಪ್ರಿಂಗ್ಸ್ F1 ಇದನ್ನ ಹೇಳಿ. gametes ಮತ್ತೆ F1 generation ಅನ್ನ ಹೇಳಿ, because the sensor is not working. I'm also getting some confusion here. XW plus XW, XW plus Y. Please. I want F1 generation ಅಲ್ಲಿ ಏನೇನು ಕಾಂಬಿನೇಷನ್ ಬರ್ತವೆ ಅಂತ. F1 generation. Let me know. Take your 2 minutes of time and let me know. Try it. Okay, Priyanka answered it. So here what happens in the F1 generation, all we got is all we got is red-eyed color. So in F1 generation, yeah, Ishu also cleared it. So XW plus, fine. So all we got is red-eyed color. Then again, now in the F2 generation ಅಲ್ಲಿ ಏನ್ ಮಾಡ್ತೀವಿ ಈಗ ನೋಡಿ, F2 generation ನಾವ್ ಏನ್ ಕನ್ಸಿಡರ್ ಮಾಡ್ತೀವಿ, ಪ್ಲೀಸ್ ಡೂ ಇಟ್. XW plus I'm sorry. Okay. XW plus XW plus crossed with XW, okay, XW plus, this is a female, crossed with XWY. Yeah. Do it and let me know. Sorry, it is XW plus Y. It is XW plus Y. This is a female. This is from F1 only. F1 generation we took. So after crossing, we get here how many gametes? XW plus. This is one gamete. XW. This is other gamete. Here also XW plus one gamete. Here also Y. This is Y. Okay. So when we do the crossing, one we got XW plus XW plus. Okay. Then again, XW plus Y. We got XW plus Y. Okay. Next we got XW plus XW. Next we got XWY. XWY. So when you observe the F2 generation, guys, XW plus XW plus, is it a male or a female? This is a female with red-eyed. This is a female with red-eyed. This is a male with red-eyed. This is a male with white-eyed. So observe carefully. Anywhere did you get female with the white-eyed? Anywhere did you get female with the white-eyed? Yes, all of you. Fine. So this indicates that whatever the eye color is present, is present on the X-linked gene, X chromosome. See here, XWY anta bandaga. So that white color or eye color pertaining, character ge pertaining color enide. So that gene is on the X chromosome. So one more student is present. The name is as iPhone anta. What is your name? Please let me know your name. So by this, the Morgan confirmed by the monohybrid cross in the ಅವ್ರೇನ್ confirm ಮಾಡಿದ್ರು ಅಂತ ಅಂದ್ರೆ the X-linked inheritance ಅನ್ನ propose ಮಾಡಿದ್ರು. That is, the eye color is present on X-linked inheritance. Next, one more we have dihybrid cross ಅನ್ನ consider ಮಾಡ್ತಾರೆ. ಯಾರು? Morgan ಅವರು. Dihybrid cross. So we know that in the dihybrid cross, we will consider how many characters? 2 characters. How many characters? 2 characters. So what he did first in the dihybrid cross, one second, I will just confirm. Yellow bodied, white-eyed. White-eyed female. Okay. So yellow bodied, white-eyed female is crossed with brown bodied, red-eyed male. Brown bodied, red-eyed male. So yellow body, yellow comma, this is a body color, yellow body, white-eyed female. White-eyed female anna he crossed with brown body, red-eyed male. So this is a male one. Out of which this is a dominant type or this is a wild type. Brown ಮತ್ತೆ red ಅನ್ನೋದೇನಿದೆ, brown body red dyed ಅನ್ನೋದು, that is a dominant type. So let us consider it as YW plus Y plus W plus ಅಂತ. Okay, then this as YW ಅಷ್ಟೇ. The genotype as YW and this as Y plus W plus. Okay, so during the gamete formation we will get Y, we will get W here, and here we will get Y plus, we will get W plus. Please cross it and let me know. These are the gametes, guys. Okay, please cross it and let me know. YW, Y plus W plus. So what all combination we get? So one ನಿಮಿಷ. This is a dihybrid cross alva. Dihybrid cross. Okay, please rectify. YW consider this yellow bodied white eyed female. Okay, crossed with Y plus. I'm sorry for the technical issue, guys. ಏನಾದ್ರೂ ಬರೆಯಕ್ಕೆ ಹೋದ್ರೆ ಡಿಲೀಟ್ ಆಗ್ತದೆ. Brown bodied red eyed male. Okay, I should please rectify it. Fine. So we got the gametes as YW here and Y plus W plus here. Y plus W plus here. So when we cross, what we will get? Let me know, please. When we cross this, what all we got? Y plus Y, W plus W. So Y plus anta andre brown. It is a dominant one. So this is dominant. This is recessive. Brown. Then W plus andre red. So all we got is brown and red. Yeah. No, dihybrid cross, Priyanka. Dihybrid cross. I changed it. Dihybrid cross. Now monohybrid cross tara solve ಮಾಡ್ತಾ ಇದ್ದೀವಿ. This is a dihybrid. Please do observe the whiteboard. Yeah. Take your time and do. All of you take your time and do. ಎಲ್ರು ಬರಿತಾ ಹೋಗಿ. So this is actual what we should get in the dihybrid cross. But when Morgan observed, he got wild type, that is brown bodied red eyed color, and one more he got is yellow color with white eyed. Yellow bodied with white eyed siktu, brown bodied with this one white red eyed siktu in a heterozygous condition. After that, in the F2 generation ಅಲ್ಲಿ ಏನಾಯ್ತು ಅಂತಂದ್ರೆ 98.7%, when we considered 100%, 98.7% he got it as from the parental type, that is brown bodied red eyed wild type, brown bodied red eyed. Whatever we got 1.3%, 100% ಅಲ್ಲಿ 98.7% parent ತರ ಆಯ್ತು. ಉಳಿದಿರುವ 1.3% ಏನ್ ಆಫ್ ಸ್ಪ್ರಿಂಗ್ಸ್ ಸಿಕ್ತಲ್ವಾ, ದ ಆಫ್ ಸ್ಪ್ರಿಂಗ್ಸ್ ವಿಚ್ ವಿ ಆಬ್ಟೇನ್ಡ್, ದ ರಿಮೈನಿಂಗ್ 1.3% ವೇರ್ ಡಿಫರೆಂಟ್ ಫ್ರಮ್ ದ ಪೇರೆಂಟ್ಸ್. ದಟ್ ಈಸ್ ದೇ ಮೈಟ್ ಬಿ ಹ್ಯಾವಿಂಗ್ ಯೆಲ್ಲೋ ಬಾಡಿಡ್ ರೆಡ್ ಐಡ್. ಓಕೆ, ಲೈಕ್ ದಟ್. ಯೆಲ್ಲೋ ಬಾಡಿ ರೆಡ್ ಐ ಆಗಿರಬಹುದು, ಅಥವಾ ಬ್ರೌನ್ ಬಾಡಿ ವೈಟ್ ಐಡ್, like this different ಆಗಿರೋದು ಸಿಕ್ತು from the parents. That is only 1.3%. So actually, can anyone tell me the dihybrid cross, the phenotypic ratio of dihybrid cross? Phenotypic ratio ಏನು? The normal phenotypic ratio of dihybrid cross. Normal dihybrid cross phenotypic ratio. You learned it, alva? Phenotypic ratio of monohybrid cross is 3:1. So phenotypic ratio of dihybrid cross. Yeah, please. Be fast. Very good. Aishu responded 9:3:3:1 anta. But according to Morgan, so when he solved, idu dihybrid cross, Mendel avurdu naveeniga madiddu. But when he observed experimentally, he did not get 9:3:3:1 dihybrid cross alli. He did not get this ratio. He obtained different ratio in the F2 generation. Ade heldage 98.7% he got the parental type only. What was the parental type? Brown bodied, red eyed was a wild type. Yeah. So was a wild type alva. So ade siktu avrige. So this disproves the independent assortment. Idenagate law of segregation anna idu this will not andre opkolodilla. So according to Mendel, what factors will segregate anta heltare alva? Factors will segregate anta heltare. But here the segregation was not proved. So what he says, Morgan says that according to Morgan, what he says is on the X chromosome, the white-eyed color was present. So most of the characters which are linked to the sex or which are linked to the eyes and all, they are on the X chromosome. So crossing adre, there might be a chance of recombination. So X chromosome mele jaasti genes irodrinda, so we got like this anta he proved. So that is why Morgan is considered as a father of experimental genetics. I hope it is clear. Fine. So let us proceed with the next concept here. Now we have polygenic inheritance. Very interesting concept here. Polygenic inheritance. Poly means what? Many. Genic means genes. So sometimes you observe whatever Sir Mendel considered. Let us consider, he considered the height of a stem. Height of a stem anna avaru consider madidru. Height of a stem anna consider madadaga he considered only two forms, tall or dwarf. No intermediate or in between condition. When he considered the color of the flower, violet and white. No intermediate conditions or the colors. But in all the characters or in all the organisms, only two forms will not be there. For example, in the human beings itself, if my father as well as my mother is tall, it is not that my tallness will be also exactly as them only. For example, if my father is 6 feet, my mother is also 6 feet, nanu 6 feet irbekanta enilvalla. So I might be 5.6, 5.4 like that. Okay. So by considering these all, one more concept one more concept came into light, that is the traits which are generally controlled by 3 or more genes. So illi namma Mendel or prakara ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ 2 genes ಇಂದ ಮಾತ್ರ ಏನಾಗ್ತಾ ಇತ್ತು, ಕಂಟ್ರೋಲ್ ಆಗ್ತಾ ಇತ್ತು. But here in most of the organism, as we will get many forms, consider this diagram, the tallness of a parent. See here, the parents are tall. So how tall is their son will be? We can't predict, because the height character or many characters will be controlled by 3 or more genes. Erade genes inda illi control agok sadhya illa most of the organisms alli. So yava character 3 or more genes inda control agatto, that character, that gene is called, that character is called polygenic character. ಆ ಜೀನ್ಸ್ ಗೆ polygenic genes ಅಂತ ಕರೀತಾರೆ. and that will transfer to the generation alva. So that is called as polygenic inheritance or quantitative inheritance. As you can see, the traits which are generally controlled by 3 or more genes and are thus called polygenic inheritance or quantitative. ಇದಕ್ಕೆ ಇನ್ನೊಂದು ಹೆಸರು ಏನಂತ ಅಂದ್ರೆ quantitative inheritance ಅಂತ. Those 3 or more genes are considered as polygenes. So in the polygenic inheritance, we have several illustrations. The first one is kernel color in wheat. Wheat ಅಂದ್ರೆ ಗೊತ್ತು ನಮಗೆ ಅಲ್ವಾ? So ಗೋಧಿ. So ಗೋಧಿಯ kernel color ಏನಿರುತ್ತೆ, the outer layer color. So that will show polygenic inheritance ante. This was put forth by a scientist named H. Nielsen Ihil and East. H. Nielsen Ihil and East. So the genetics, those persons were geneticists, okay? So what they did is they crossed a red color kerneled variety with white kerneled variety. So white color iruva wheat jote red color kernel iruva variety anna cross ಮಾಡ್ತಾರೆ. Grains of F1 were uniformly red but intermediate between the red and white of parental generation. So what they got in the F1 generation? ರೆಡ್ ಕಲರು ಸಿಕ್ತು, ವೈಟ್ ಕಲರು ಸಿಕ್ತು, ಮತ್ತೆ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ಬಿಟ್ವೀನ್ ರೆಡ್ ಅಂಡ್ ವೈಟ್ they got. So when members of F1 were self-crossed among themselves, 5 different phenotypic classes appeared in F2. So again, when these F1 generation were crossed, okay, well allowed for self-pollination, okay, self-crossing allow ಮಾಡಿದಾಗ, whatever we got in the F2 generation ಅಲ್ಲಿ 5 different varieties ಸಿಕ್ತಂತೆ ನಮಗೆ, 5 different varieties. So let us see that. See here. So parents were dark red wheat kernel and the white wheat kernel. So these are the genes AA, BB, CC. All these 3 genes will control the color of the kernel. So when all these are homozygous, the color of the kernel will be dark red in color. When all these are dominant, the color will be dark red. When all these are recessive, the color will be white color. So when there is a cross between 2, we got a heterozygous condition. So it was intermediate red ante. This was intermediate red. Andre dark red agirlilla, white agirlilla ante. Then after that, when this intermediate red is allowed for self-pollination. So ಇಲ್ಲಿ ನಾವು ಟ್ರೈ ಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್ ಮಾಡ್ತೇವೆ, ಯಾಕೆಂದ್ರೆ we considered 3 genes here alva. So when intermediate or self-pollination occurred in this, we got 64, total agi 64 plants siktu. ಆ 64 plants ಅಲ್ಲಿ 1 was dark red, 6 was moderately red, 15 were actual red, 20 were intermediate red, 15 were light red, 6 were very light red, 1 was white. See here, 63 red many shades al siktave 63, 1 irodu white. So total 63 plus 1 white, 64. Fine. So this is a kernel color. How we got to know this? This is controlled by many genes. So ಎಷ್ಟು amount of what we can say dominant alleles irtavo, that much color will be there. For example, in this table you can observe, ಎಲ್ಲಾದ್ರೂ ಡೌಟ್ ಬಂದ್ರೆ ಅವಾಗ್ಲೇ ಕೇಳಿ, don't hesitate, ಹೇಗೆ ಕೇಳಿ ಅಂತ. Just a minute. Okay, observe this one. Parents were dark red, so they consider R1, R2, R2 dominant one, and this is white. Whatever they got in F1 generation was intermediate athwa medium red. F2 generation alli ಏನು ಸಿಕ್ತು ನೋಡಿ, all are dominant here, R1, R2, R2. So it is dark red. So here in the second variety, ಇದು 4 ಅಂದ್ರೆ 4 plants ಈ ರೀತಿ ಸಿಕ್ಕಂತೆ. So R1, R2, R2. So here number of dominant alleles are more. So it is medium dark red anta. When you consider this one, so here R1, R2, R2. Here we have only 2 dominant alleles. So it is medium red. Next, when you consider this R1, R1, R2, R2. So only one dominant allele is present. So it is light red. So at last we got all the recessive alleles. So white color siktu. As you observe, the number of dominant alleles in ಮಾಡುತ್ತೆ, it influences the color of the kernel. The number of dominant alleles influences the color of kernel. If the dominant alleles are more, the red color or the red shade will be more. Okay. By this, we will understand that certain characters in many organisms will be controlled by 3 or more genes. Did you get, guys? ಅರ್ಥ ಆಯ್ತಾ ಎಲ್ರಿಗೂ? Yes. Respond. Please respond. Okay, thank you. Then again, one more example we have in the polygenic inheritance itself. Just a minute. Yeah. That is skin color in human beings. So skin color, ಒಂದು ತೀಪರಿ ವೈಟ್, ಇನ್ನೊಂದು ತುಂಬಾ ಡಾರ್ಕ್ ಅನ್ನೋ ಮ್ಯಾಟರ್ ಇಲ್ಲ. So we can see variety of skin colors in the human beings. See here, the presence of melanin pigment in the skin determines the skin color. How much melanin is present in our skin. So that determines the skin color ante. The amount of melanin developing in the individual is determined by 3 or 2 pairs of genes. So ಎಷ್ಟು amount of melanin ನಮ್ಮಲ್ಲಿ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಬೇಕು ಅದನ್ನ ಕಂಟ್ರೋಲ್ ಮಾಡೋದು 3 or 2 pairs of genes ಅಂತೆ. These genes are present at 3 different loci. ಒಂದೇ ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿ 3 different position ಅಲ್ಲಿ ಈ ಲಿಂಕ್ಸ್ ಈ genes ಇರ್ತವೆ. And each dominant gene is responsible for synthesis of fixed amount of melanin. Amount of melanin produced is always proportional to number of dominant genes. Okay? So, ಎಷ್ಟು melanin produce ಆಗಿದೆ ಅದು ಯಾವುದರ ಮೇಲೆ ಆಧಾರವಾಗಿರುತ್ತೆ ಅಂದ್ರೆ how many dominant genes we have. Based on that, the skin color and melanin amount will be there. See here, the frequency, this is a number of dominant alleles. ಇಲ್ಲಿ ನೋಡಿ ಈ ಒಂದು ಇಮೇಜ್ ಅನ್ನ ನೋಡಿ. See all this one alleles here in this condition. Can you observe 0 ಇದೆ ಇಲ್ಲಿ reading. In this all the alleles are recessive condition. See the color of the girl. She is white. Means melanin frequency amount kadme irutte recessive iddaga. When you see this A, B, B, C, see here in this example you can see one dominant allele is present. One dominant allele is present. So ಅವರು ಇಲ್ಲಿಗೆ ಬಂದಿರ್ತಾರೆ, this color. Okay, next when you consider these all, so ಇಲ್ಲಿ the number of dominant alleles as you are seeing, no, they are 2 to 3 dominant alleles ಕಾಣಿಸಿಗತ್ತೆ ಇಲ್ಲಿ. So though in these people the melanin pigmentation, the amount of melanin produced will be more. So ಇಲ್ಲಿ ನೋಡಿ ಡಾಮಿನೆಂಟ್ ಅಲೀಲ್ಸ್ ಏನಿವೆ ದೇ ವಿಲ್ ಆಡ್ ದ ಮೆಲಾನಿನ್. ಮೆಲಾನಿನ್ ಜಾಸ್ತಿ ಆಡ್ ಆದಂಗೆ ನಮ್ಮ ಸ್ಕಿನ್ ಕಲರ್ ಡಾರ್ಕ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಓಕೆ, ದೆನ್ ಆಫ್ಟರ್ ದ್ಯಾಟ್, ಸೊ ವೆನ್ ಯು ಸಿ ಫ್ರಮ್ ದ ಫಿಫ್ತ್ ಒನ್ ಹಿಯರ್, ವೇರ್ ಇಸ್ ದಟ್ ರೀಸನ್, ಯಾ, ಫ್ರಮ್ ದ ಫಿಫ್ತ್ ಸ್ಕೇಲ್, see here more than 3 or 4 dominant alleles are present. See the skin color. At last all the dominant alleles are present. See the skin color, right? So like this, how many dominant alleles will be present, that much effect will be there. If the dominant alleles are more, addition of melanin will be more to the skin color, then we will be dark. If the amount of if the alleles are recessive, if the only few dominant alleles are present, alleles andre genetic forms, then less addition of melanin takes place, then we will be white. Understood this, all of you? Understood, all of you? Bega, please respond. Okay, thank you. Okay, so in a polygenic inheritance related to NEET and CET, we will be getting some questions. ಯಾವ ರೀತಿ ಅಂತ ಇಲ್ಲಿ ನೋಡಿ, calculate the sum total of phenotypes and genotypes in F2 generation by 2 pairs of polygenes. So ಈ ರೀತಿ ಕೇಳಿದಾಗ sum total of phenotypes ಮತ್ತೆ genotypes ಅಂತ ಕೇಳಿದಾಗ, see here 2 pairs ಅಂದ್ರೆ 4 genes ಅಲ್ವಾ. see here, the number of phenotype for polygenes ಅಂತ ಅಂದಾಗ 2n plus 1. n is what here? n is the number of pair of genes. So 2 2s are 4, 4 plus 1, 5. So number of genotype ಅಂತ ಕೇಳಿದಾಗ 3 raised to 2. So that is 9. So ಇಲ್ಲಿ ಚೆಕ್ ಮಾಡೋಣ ನೋಡಿ. ಇಲ್ಲಿ 3 ಜೀನ್ಸ್ ಏನಾಗಿದೆ, 3 ಪೇರ್ ಆಫ್ ಜೀನ್ಸ್ ಏನಾಗಿದೆ, ಇಲ್ಲಿ ನಮಗೆ 3 ಪೇರ್ ಆಫ್ ಜೀನ್ಸ್ ಇದೆ ಕರ್ನಲ್ ಆಫ್ ವೈಟ್ ಕಲರ್ದು. ಸೊ ನಂಬರ್ ಆಫ್ ಫಿನೋಟೈಪ್ ಎಷ್ಟು? 2 plus 3 so 2 into 3 is 6. 6 plus 1 is 7 ಫಿನೋಟೈಪ್ ಸಿಗತ್ತೆ. ಅದೇ ರೀತಿ ನಂಬರ್ ಆಫ್ ಜೀನೋಟೈಪ್ ಫಾರ್ ಪಾಲಿಜೀನ್ಸ್ ಅಂದ್ರೆ 3 raised to 3 pairs ಇದೆ, ದಟ್ ಈಸ್ 9. Okay, fine. So like this, we have to calculate the polygenic inheritance. Based on this, we will get the problems. Artha aita? Is it clear, guys? Okay. Dark skin color. Which I have to explain, Purnima? ಫಿನೋಟೈಪ್ ಅಂದ್ರೆ ಫಿಸಿಕಲಿ ಎಷ್ಟು ಸಿಗತ್ತೆ ನಿಮ್ಗೆ ಅಂತ, ಫಿಸಿಕಲಿ ಎಷ್ಟು ವೆರೈಟೀಸ್ ಸಿಗತ್ತೆ ಆಫ್ ಸ್ಪ್ರಿಂಗ್ಸ್ ಅಲ್ಲಿ. ಇಟ್ ಇಸ್ ಅ 2n plus 1. ನಂಬರ್ ಆಫ್ ಜೀನೋಟೈಪ್ ಎಷ್ಟು ಸಿಗ್ತವೆ? n ಅಂದ್ರೆ ನಂಬರ್ ಆಫ್ ಪೇರ್ ಆಫ್ ಪಾಲಿಜೀನ್ಸ್. n ಮೀನ್ಸ್ ನಂಬರ್ ಆಫ್ ಪೇರ್ ಆಫ್ ಪಾಲಿಜೀನ್ಸ್. ಇಲ್ಲಿ ನಾನು ಪ್ರಾಬ್ಲಮ್ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿಲ್ಲ ಅಮ್ಮ. ಪ್ರಾಬ್ಲಮ್ ಹೇಗ್ ಸಾಲ್ವ್ ಮಾಡ್ಬೇಕು ಅನ್ನೋದನ್ನ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೇನೆ. ಪ್ರಾಬ್ಲಮ್ಸ್ ಅನ್ನ ಕ್ವಶನ್ ಸಾಲ್ವ್ ಮಾಡುವಾಗ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೇನೆ. ಸೊ ಇಲ್ಲಿ ನಂಬರ್ ಆಫ್ ಫಿನೋಟೈಪ್ ಫಾರ್ ಪಾಲಿಜೀನ್ಸ್, ಎಷ್ಟು ಫಿನೋಟೈಪ್ ಗಳು, ಫಿಸಿಕಲ್ ವೆರೈಟೀಸ್ ಗಳು ಸಿಗ್ತವೆ ಅಂತ ಅಂದ್ರೆ 2n plus 1. ನಂಬರ್ ಆಫ್ ಜೀನೋಟೈಪ್ ಫಾರ್ ಪಾಲಿಜೀನ್ಸ್ ಅಂದಾಗ 3 raised to n. n ಅಂದ್ರೆ ಏನ್ ಮೇಡಮ್ ಅಂತ ಅಂದ್ರೆ ನಂಬರ್ ಆಫ್ ಪೇರ್ ಆಫ್ ಪಾಲಿಜೀನ್ಸ್. ಫಿನೋಟೈಪ್ ಅಂದ್ರೆ ಫಿಸಿಕಲಿ, ಜೀನೋಟೈಪ್ ಅಂದ್ರೆ ಜೆನೆಟಿಕ್ ಮೇಕಪ್. ಡಿಡ್ ಯು ಗೆಟ್? The movement of a gene from one linkage group is a translocation. Okay. Answer to these questions now. Select the correct statement from the ones given below with respect to dihybrid cross. Anta ide. Tightly linked genes on the same chromosome show higher recombination. Genes far apart on the same chromosome show very few recombination. Genes loosely linked on the same chromosome show similar recombination. Tightly linked genes on the same chromosome show very few combinations. What is the correct statement with respect to dihybrid cross? Priyanka answered as option A. What about others? ನೀಟಾಗಿ ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ. ಜಸ್ಟ್ ನಾವು ವಿ ಡಿಸ್ಕಸ್ಡ್ ರಿಗಾರ್ಡಿಂಗ್ TH Morgan's experiment. ಟೈಟ್ಲಿ ಲಿಂಕ್ಡ್ ಜೀನ್ಸ್ ಅಂತಂದ್ರೆ ತುಂಬಾ ಕ್ಲೋಸ್ ಆಗಿ ಅಸೋಸಿಯೇಟ್ ಆಗಿರುವಂತಹ ಜೀನ್ಸ್. ರೀಕಾಂಬಿನೇಷನ್ ಅಂತ ಅಂದ್ರೆ ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಆಗೋದು. ಸೊ ಒಂದೇ ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿ ಜೀನ್ಸ್ ಗಳು ಟೈಟ್ಲಿ ಲಿಂಕ್ ಆದಾಗ ರೀಕಾಂಬಿನೇಷನ್ ಜಾಸ್ತಿ ಇರತ್ತಾ ಕಡಿಮೆ ಇರತ್ತಾ ಅಂತ ಕೇಳಿದ್ದಾರೆ ಅವರು. That's why while explaining you have to write down the few points. ಸುಮ್ನೆ ಟಿವಿ ತರ ನೋಡ್ತಾ ಕುತ್ಕೋತೀರ. ಟೈಟ್ಲಿ ಲಿಂಕ್ಡ್ ಜೀನ್ ಸೇಮ್ ಕ್ರೋಮೋಸೋಮ್ ಇದ್ದಾಗ ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಆಗಕ್ಕೆ ಹೇಗ್ ಸಾಧ್ಯ? ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಆದ್ರೆ ರೀಕಾಂಬಿನೇಷನ್ ಆಗತ್ತಲ್ವಾ? ದೆನ್ ಆಪ್ಷನ್ ಡಿ is a right answer here. Next question. Inheritance of blood groups in humans. It is an example of dominance, codominance, multiple allele, incomplete dominance, polygenic inheritance. Option A, 2, 3, 5 are right. Option B, first, second, and third are right. Option C, 2, 4, and 5th is right. Option D, 1, 3, and 5th is right. Yeah. Inheritance of blood groups in humans shows which all properties? Dominance, which all mechanisms or the phenomena? Dominance, codominance, multiple alleles, incomplete dominance. Be fast. Be fast. AB blood group shows codominance. Aishu answered it correctly. Option A, 2, 3, and 5. Multiple allelism. Then 2, codominance, as well as polygenic inheritance. Okay. Inheritance of skin color in humans is an example of point mutation, polygenic inheritance, codominance, chromosomal aberration. Two, three, four, two, three, four, two, three. Two, 4, 5, 1, 3. Priyanka answered it. Option B, polygenic inheritance is the right answer. Next question. Select the incorrect statement. Human males have one of their sex chromosome much shorter than other. Male fruit fly is heterogametic. This is not explained. Innu explain madilla. You sex linked inheritance athwa adral baratte. This I will be explaining in the next session. Okay. Fine. Okay. Let me end up the session here. Have a nice day. Take care. Bye bye.